Okay, so grade 6 girls, we will continue the lesson DNA and its function. And yesterday you have seen the video where I've shown how step by step the DNA is copied, okay?
Okay.
And that is when they copy before the cell division. Anytime the cell divide, from the day one when I teaching you cell, I told you when the cell divide, before it splits into two nucleus, or before the cell divides, the DNA copy. So that copies of DNA, the process is called as replication.
Replication.
But during the replication, two strands, they separate almost like through threads. That's the first thing. The DNA molecule open up, separating into two strands. This, what you call as diagram is very important, girls. The DNA molecule open up, separating into two strands. Clear? Second point. Nucleotides match up. Nucleotides match up and join the open DNA strand. These nucleotides, they match up and join to the open strand. They try to make them together and they come and join. And in the C, finally, it becomes joined. Identical, two identical molecules are formed. Two identical molecules are formed. Do you understand the diagram now? First, the DNA molecule open up. Whose mic is that disturbing you?
Me.
Aline, you can mute it. Dear.
Okay.
But that doesn't mean, if I ask you a question, I'm sure you are answering always, okay?
Okay.
Okay, good. So the DNA molecule opens up, separating into two strands. And here, because of nucleotide, when they separated two strands, because of nucleotide, they match up and join to the open DNA strand. They match up, they come close and join. And finally, two identical molecules are formed. Is this clear? Coming to me.
Yeah.
Changes in the number type or order of the bases on a piece of DNA are known as mutations. Girls, we will just highlight few things here. Changes in the number type of order or bases. You remember I told you there is one base, one sugar, and one phosphor. You remember that? So if there is some change in the number, it is called as mutation. Sometimes a base is left out. This kind of change is known as deletion. Sometimes a base is left out and we need to delete that base, and that is called as deletion. They don't pair up. You remember the bases? There were 4 bases: adenine, guanine, thymine, and cytosine. Remember?
Yeah.
Okay. So if it is some base is left out, that has to be kicked down. That is called as deletion. Sometimes an extra base might be added. That is called as insertion. The most common mutation happens when one basis replaces another, and that is substitution. The most common mutation happens, the whole para, my dear, when one base replaces another. This kind of change is known as substitution. Okay?
Okay.
So again, if one goes out, left out, deletion. Added extra, insertion. Replaces one another, it's called as substitution, okay?
Okay.
Please count these.
This is the function of the substitution.
We will check, we'll count and check. Count the first original sequence. 1, 2, 3, 4, 5, 6, 7, 8, 9. Count A, 3, 6, again 9. See, in the B1, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. You have 10. You have one extra in B. And how about in C? Check, girls. How about in C? 1, 2, 3, 4, 5, 6, 7, 8. Okay. In 8, it is left over is in the first one. In the second, it is extra. Come on, tell me. A, what do you think the answers are? Tell me.
Tell me.
A, it's same, girls, same number. So that means it's replaced. What is the answer for the A?
Replace.
Replace, because it's same number. 9 and 9. That means it is substitution.
Substitution.
Yes. And tell me 1, 2, 3, 4, 5, 6, 7, 8, 1, 2, 3, 4, 5, 6, 9, 10. Okay. And
Insertion.
insertion.
Insert.
And D is deletion because you deleted.
Yeah.
Okay.
Okay.
Understood, girls? The concept?
Yeah. Miss. Yeah.
Okay. How do mutations happen given the large number of bases in an organism? DNA, it's not surprising that random error can occur during replication. Some errors can occur, but sometimes DNA can also be damaged by some physical and chemical agents called as mutagens. Girls, you know the UV rays which come from the sun, they are very, very harmful, but they don't reach us. They don't reach us because there is one layer called as ozone layer in the sky, in the atmosphere. There is a layer that's called as ozone layer in the atmosphere that doesn't allow ozone to reach. Imagine, girls, the sun could have given that UV rays. You know, we couldn't see the yellow color. It would be strong purple color. The whole earth, we will not have that sunlight. It will have strong purple color.
Purple?
Yes, UV rays are purple in color. Can you imagine? So girls, that's why the DNA can be damaged by those mutagens. Mutagens means there are UV rays and some chemicals in cigarettes. Those people who drink cigarette, they can get the problem of DNAs. Girls, will you give me a minute? I will come back. All right, so we did that. The first one is substitution because the same number, that means it has been replaced. The second one, it's insertion because they are extra. And in the third one, it's deleted. Okay? Now, the snake, and I told you what can alter, girls? Mutagens. Mutagens can alter. That means that can affect DNA, that can harm DNA. And mutagens are such as UV rays, that is ultraviolet rays that comes from the sun, and sometimes the substance present in the cigarette. Cells make proteins that can fix errors in DNA, but sometimes a mistake is not corrected and become part of genetic code. Mutations to DNA may be beneficial. Sometimes they are good, sometimes they are neutral, or sometimes they are harmful. A genetic disorder results from mutations that harm the normal function of the cell. The cell cannot function properly. Like example, we have Tay-Sachs disease. We have sickle cell anemia. Okay, we have sickle. You know, in sickle cell anemia, the person blood will not be made. I'll show you both of the YouTube on the video. I'll show you the videos because you must know what are the DNA disorders. We'll see about Tay-Sachs disease and we'll also see about sickle cell anemia. Maybe if the father or the mother or the grandparents have this disease, automatically one of the baby or one of the grandchildren, they might have this disease without any reason because their grandfather had it. They will have this disease. Tay-Sachs disease, sickle cell anemia, these are inherited, passed from one parent to offspring. Okay, and even cancers, girls, even cancers come from sometimes, not sometimes, like 60% of cancer cells are because of genetic disorders. Now, what did I say you? You have to know about, I will show you sickle cell anemia. Anemia, you know, when I say anemic, that means the blood cannot make iron. I have Tay-Sachs disease, okay? Tay-Sachs disease, and I want to say the baby of the Tay-Sachs disease. Come on. Tell me if you...
Tay-Sachs disease, or TSD for short, is a lysosomal storage disorder caused by a mutation in a gene on chromosome 15, which codes for a lysosomal enzyme called beta-hexosaminidase A, or
It
Hex
is
A
very
for short.
tough for you. Don't
This
go in
enzyme
detail.
normally...
Just have a normal idea, okay? It's something in chromosome 15, something goes wrong, okay, during the mutations. And that's why the Tay-Sachs disease is there. I'll show you more and then we'll switch to the sickle cell anemia.
breaks down a lipid called GM2 ganglioside. GM2 is found mainly in neurons. So without Hex A, it accumulates inside lysosomes. TSD is also known as GM2 gangliosidosis type 1. This results in progressive symptoms.
No, I just want to show you the symptoms.
For
What
adult
happens?
onset, there may be motor difficulties, and some adults may manifest in
I
some.
want to show you what happened to the baby.
Depending on age of onset, TSD can be infantile, with onset at 3 to 6 months, juvenile, with onset at 2 to 5 years, chronic, with onset in the first or second decade of life, and late onset, with the first indication of symptoms in the second to third decade of life. Common signs for the first 3 forms are signs of CNS degeneration, like decreased muscle tone, abnormally increased reflexes, seizures, and visual disturbances.
Seizures.
For adult onset,
Yes, epilepsy, that, you know, the muscles are very, what you call as tight. The muscles cannot move. The muscle tone is very, very poor. And visual disturbances, that means the person cannot see properly. Okay? Suddenly, it starts from 3 to 6 months, or sometimes 2 to 5 years, or sometimes when they are 20, above 20 years, sometimes adults above 40 years, okay? So I just wanted to show you that Tay-Sachs disease, what are the symptoms here?
there may be motor difficulties, and some adults may manifest bipolar type psychological symptoms. Ophthalmologists may be the first to consider TSD by finding a cherry red spot in the macula.
Ophthalmologist is the doctor of eye, ophthalmologist. This is something extra information for you. You know, sometimes, as I told you, visual disturbances, there will be a red spot on the cornea, red spot, the dark red spot
Of the eye, which results from GM2 buildup in the retinal cells around the central macular area. Diagnosis of TSD is done by determining the activity of Hex A in serum, leukocytes, tears, or any other body tissue. Genetic testing for Hex A gene mutations and sequencing of the Hex A gene are used for diagnosis
They
and
just take
heterozygous carrier
the part
detection.
of the body, either they take the blood sample or they take the tears of the baby or the body tissue, and it goes and find it out, okay? This was about Tay-Sachs disease. I think I just want to have an idea, that's it. It's not in the syllabus, but you must know. Sickle cell anemia.
In people with sickle cell disease, their red blood cells become deformed, turning sickle-shaped. The bone marrow tries to make more red cells to make up for the loss, but can't keep up, causing anemia. Their new shape keeps them from moving properly through the body, and the misshapen cells can jam up and stick to the walls of the blood vessels. These clumps cut off oxygen to healthy tissue, delaying a child's normal growth and causing fatigue. and extreme pain.
Understood, girls?
Yeah, miss.
All right. Let us come back to the lesson now. What is the role of DNA and RNA in building proteins? Okay, imagine that you are baking cookies. You have a big cookbook that contains the recipe. If you take the book with you in the kitchen, you will risk damaging the book and losing important instructions, right? Sometimes you might, yes, you might make your book, and you can spill water on that or something wrong with the book. So you only need one page from the book. So you copy the recipe on a piece of paper and leave the cookbook on the shelf. Am I correct? This process is similar to the way that cell uses DNA to build proteins. First, some of the information the DNA is copied to separate molecule called as ribonucleic acid. They just copy the information. The DNA copy the information to RNA. Then the copy is used to the, and that copy, that RNA is used to build the protein. They don't take the whole DNA. You understand how they compared? How they compared to the recipe and how did they do? The DNA copy the information. It's writing down the information to a separate molecule called as RNA. Then this copy is used to build proteins. Of course, not all instructions are needed at that time. The DNA is protected inside the cell's nucleus. You know very well that DNA is inside the cell's nucleus. Now, like DNA and RNA has a sugar phosphate backbone. You know the base? It has sugar, it has phosphate, and one backbone that is a base. And the bases, what are the bases? Adenine, guanine, cysteine, cytosine. But instead of thymine, RNA contains the base uracil. Even same like DNA, the RNA has sugar, phosphate, and base AT, AGC. T is not there. Adenine is there, guanine is there, and cytosine is there. Or adenine, guanine, and cytosine. But no T, no thymine. Instead of thymine, RNA has the base uracil. What is it? The base uracil. Now, example, if I ask you DNA, you will say it's a nucleotide that consists of sugar, phosphate, bases like AT, adenine, thymine, CG. That is cytosine, and G is guanine. But in RNA, that is ribonucleic acid, you have everything same except T, except thymine. Except T, you have uracil. So also the sugar found in RNA is different from the one in DNA. There are 3 types of RNA: messenger RNA, ribosomal RNA, and transfer RNA. Each type has special role. Again, we have 3 types of RNA. One is the messenger, one is the transfer, and one is the ribosomal. There are 3 types of RNA. Did you understand?
Yeah, miss.
Yeah. Same, exactly. DNA and RNA are exactly the same. What is the difference? In T, instead of thymine, we have the base called as uracil. We have the base called as uracil. Understood? Now, let me show you the video first, and then we'll come back to that.
DNA doesn't always get copied accurately. Random errors occur, or DNA that is damaged by UV light, chemicals, or other agents can experience copying errors. Cells produce proteins that can fix some errors in DNA. However, sometimes a mutation occurs that is not fixable. A mutation is any change in the bases of a DNA molecule. Some mutations have no effect on a cell, and others can be beneficial. But some mutations can cause diseases. A genetic disorder results from mutations that affect the normal function of a cell. Some of these disorders, such as Tay-Sachs disease and sickle cell anemia, are inherited or passed on from parent to offspring. Other genetic disorders result from mutations that occur during a person's lifetime. Most cancers fall into this category. Click the pictures to learn about the different types of mutations. A deletion is a mutation in which at least one base pair is removed. A deletion is like removing letters from the middle of a sentence. A deletion, like any other type of mutation, can cause a gene to code for the wrong protein. An insertion is a mutation in which at least one base pair is added. An insertion is like adding extra letters into the middle of a sentence. A substitution is a mutation in which a base pair is replaced with a different base pair. A substitution is like changing a letter in the middle of a sentence. Substitutions are the most common type of mutation. Sickle cell anemia is caused by a substitution mutation.
See the substitution mutation? That is where the sickle cell anemia is caused.
Proteins determine a variety of traits and act as triggers for many cell processes. DNA provides the instructions for making proteins. But since DNA cannot leave the nucleus, it needs other molecules to actually build proteins. RNA, or ribonucleic acid, helps with the process of protein building. RNA is very similar to DNA with a few subtle changes. One change is that RNA contains the base uracil, U, instead of thymine.
So did you see the major difference between RNA and DNA? What is the major difference? Only the base thymine. Instead of base thymine, the base uracil is there. Okay, girls?
Okay.
In RNA, adenine, A, pairs with uracil, U. The first part of the protein building process is called transcription. During transcription, a single-stranded molecule called messenger RNA, or mRNA, copies instructions from DNA and moves these instructions out of the nucleus and into the cytoplasm. After transcription, an mRNA molecule leaves the
This
nucleus.
transcription tomorrow I'm going to explain you. I don't think so today. Tomorrow I'll finish up the lesson, girls. And then we have Wednesday. Yeah, weekend. Inshallah, we'll finish up. Inshallah, next week. Okay, take care, girls. Have a nice day.
Okay, miss.